Among the deeper mechanisms that define how cells age, few are as fundamentally misunderstood or as potentially reversible as the process known as epigenetic drift. Epigenetics refers to chemical tags, such as methyl groups, that sit on DNA and histone proteins and regulate which genes are turned on or off. These tags don't alter the underlying DNA code, but they profoundly influence how the code is read. As we age, our epigenome becomes increasingly disordered. Some genes that should be silenced become inappropriately activated, while others that should be expressed fall silent. This pattern of disorganized gene expression, called epigenetic drift, has been strongly associated with aging and age-related diseases. But crucially, this drift is not a mysterious, unchangeable function of time. It is deeply tied to the availability of specific nutrients that serve as methyl donors for the body's epigenetic machinery. From this perspective, epigenetic aging is not simply a result of biological wear and tear. It is a correctable outcome of methyl donor scarcity. Central to this system is the molecule S-adenosylmethionine, SAMe, the body's universal methyl donor. SAMe is required for virtually all methylation reactions, including those that modify DNA and histones. It is synthesized in the body through the methionine cycle, which itself depends on adequate dietary intake of methionine, folate, vitamin B9, vitamin B12, and choline. Without these inputs, the production of SAMe falters, leading to reduced or erratic methylation across the genome. Over time, this methylation instability alters gene expression in ways that mirror the disordered patterns seen in aged cells. Tumor suppressor genes may be silenced. Inflammatory genes may be inappropriately activated. Repair pathways may go dormant. The result is a cellular phenotype that looks old, even in the absence of actual DNA damage or telomere shortening. This isn't just theoretical. Experimental models have shown that deficiency in methyl donors leads to epigenetic patterns indistinguishable from those in older organisms, including increased inflammation, metabolic dysfunction, and reduced regenerative capacity. Conversely, restoring these nutrients, particularly folate, B12, and choline, has been shown to reverse many of these age-like epigenetic shifts. In some studies, repletion of methyl donors in aging animals led to normalization of gene expression, improved mitochondrial function, and even extensions in health span. This is a striking reminder that the epigenome is not fixed. It is responsive, adaptable, and deeply dependent on nutritional status. The implications for human health and aging are enormous. Many age-associated diseases, including cardiovascular disease, cognitive decline, and certain cancers, are now understood to involve epigenetic dysregulation. But if this dysregulation is fueled by methyl donor deficiency, then prevention may lie not in pharmacology, but in micronutrient adequacy. Unfortunately, subclinical deficiencies in folate, B12, and choline are alarmingly common, particularly in older adults and those following restrictive diets. Even among those who appear healthy, long-term marginal intake can quietly erode the epigenetic landscape of their cells, subtly pushing them toward dysfunction. This understanding also helps explain the wide variability in how people age. Some individuals maintain sharp cognition, resilient immunity, and metabolic stability well into advanced age. Others experience premature decline. While genetics play a role, the long-term integrity of epigenetic regulation, supported by methyl donor availability, is a major determinant. Those who eat nutrient-rich diets, minimize alcohol intake, which depletes methyl donors, and avoid chronic inflammation, which disrupts methylation patterns, tend to preserve a more youthful epigenetic profile. Their gene expression remains balanced and adaptive, proof that aging is not just about what's encoded in the DNA, but how that code is expressed over time. Even more compelling is the fact that nutritional restoration can work retroactively. Supplementation with methyl donors has been shown in some studies to improve mood, reduce homocysteine, a toxic byproduct of methylation imbalance, and even influence biological age markers, such as DNA methylation clocks. These interventions don't simply slow decline. They may actually reverse some of its molecular signatures. This reinforces the idea that aging is not, at its core, a genetic problem. It is an environmental one, shaped by decades of subtle but chronic insufficiencies. The drift of the epigenome is not a surrender to time, but a consequence of unmet biochemical demand. And that demand is measurable, trackable, and most importantly, modifiable. By ensuring adequate intake of methyl donors through diet or targeted supplementation, we can restore much of the precision to the body's genetic control systems. In the end, epigenetic drift is not proof of inevitable decline. It is evidence of what happens when the body is asked to manage complex gene regulation without the nutrients required to do so. Reframing this process as methyl donor deficiency rather than biological wear grants us not only clarity, but also agency. It tells us that the language of our genes is still legible and still modifiable, so long as we give the body the resources it needs to write it properly.